Glycolysis. In the first step, glucose gets converted to glucose 6-phosphate. An ATP molecule is consumed in this reaction. Hexokinase enzyme catalyzes this reaction. In the second step, glucose 6-phosphate converts into fructose 6-phosphate. Isomerase enzyme catalyzes this reaction. This is a reversible reaction. Let's move on to the next reaction. In this reaction, fructose 6-phosphate turns into fructose 1,6-bisphosphate. Enzyme phosphofructokinase boosts this conversion. Remember, this reaction gobbles an ATP molecule. This is a reversible reaction. Let's see what happens ahead. In the next reaction, fructose 1,6-bisphosphate is cleaved into two products, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. The enzyme involved in this reaction is aldolase. Remember, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are interconvertible to some extent. Isomerase enzyme could convert them into each other. Let's jump on to the next reaction. In this reaction, glyceraldehyde 3-phosphate forms 1,3-bisphosphoglycerate. The enzyme in action in this reaction is glyceraldehyde 3-phosphate dehydrogenase. Remember, NADH is formed in this reaction, while an inorganic phosphate is consumed. Moving on to the next reaction. In this reaction, 1,3-bisphosphoglycerate forms 3-phosphoglycerate. Phosphoglycerate kinase enzyme boosts this reaction. Remember, this reaction forges an ATP molecule. In the next reaction, 3-phosphoglycerate gets converted to 2-phosphoglycerate. This time, the enzyme in action here is phosphoglycerate mutase. Let's jump to the next reaction. In this reaction, 2-phosphoglycerate forms phosphoenol pyruvate. Enolase enzyme catalyzes this reaction. Remember, a water molecule is released in this reaction. Moving on to the next reaction. In this last reaction, phosphoenol pyruvate is converted to pyruvate. Pyruvate kinase enzyme facilitates this reaction. An ATP molecule is produced in this reaction.